students try to grow cells but without an incubator. Let's see.
Want to hold it's been 3 days, please grow.
Maybe if I give it my bomb, the cells will grow. Without proper humidity, temperature or CO2, the cells would die. Our experiment failed. And then comes the hero of the modern biotechnology, the incubator.
This is a shaker incubator from the company Diagnosis 24. It is an equipment which is designed to provide a controlled environment for the growth and reproduction of microbial samples, cells, tissues, but also providing simultaneously shaking, agitation.
I'm a partner who's the multitasker in Biotech Labs. No, it's not us. It's the one and only incubator. From shaking a flask like a DJ's to keeping the microbes cozy at the perfect temperature.
And making sure oxygen parties with your cultures all night round. This machine does it all. Without it, imagine manual shaking flask 24/7. Biotech Labs would literally turn into gym. So next time, if you see an incubator, just remember, it's not just the science, it's the ultimate entertainer of the lab. This is the display in the shaker incubator. It shows the current time and the date and the program number. Shows the current status value, that is actual RPM. RPM is in revolutions per minute. It also shows temperature, that is 30 degrees Celsius in here. Remaining time is 0 minutes. And the system condition is power on.
Now we'll see the inside system of an incubator. Let's see.
The glass door helps maintain sterile and stable environment inside. Walls are made up of polished stainless steels just to prevent rusting. Below we can see conical flask with cotton plugs. These allow microbes to grow in liquid media. The shaker tray moves continuously, keeping the culture well mixed. At the back there is a fan for even temperature distribution. All these systems work together to provide a controlled environment for microbial growth and experiments.
Incubators are the backbone of biotech experiments. They are used for cell culturing, for the rapid growth of the microbial cells, and for microbiology to cultivate the cells, and in molecular biology. It is also used in fermentation process for IVF and also for vaccine production and in pharmaceutical industry for the drug discovery.
It provides a perfect controlled environment of temperature, humidity and CO2 for cell cultures and microbial growth. यार अब मैं ही इंक्यूबेटर बन चाहती हूँ। चिल कर रहा था, इंक्यूबेटर है ना।
We are introducing CO2 Incubator, our lab's ultimate support system. Ours is from thermoscientific. Basically, it's like a 5-star hotel where mammalian cells like to chill at 37 degrees Celsius. It is used in cell culture experiments, from cancer research to vaccine development, tissue engineering, and drug testing. Anytime you hear words like stem cells, antibodies, trust me, those little guys were partying in the CO2 incubator first. CO2 incubator works on 5 principles. First, it maintains the temperature 37 degrees Celsius just like a human body. Second, it maintains 5% CO2 inside the chamber. Third principle is the CO2 reacts with the bicarbonate in the medium, keeping the pH perfectly blended for the cells. And the fourth principle is a watery tray inside ensures high humidity so cultures don't dry out. So it's like creating a mini human body environment so cells feel right at home.
Now, let's take a look to our CO2 incubator. Normally, we don't talk directly in front of it because it's placed inside the laboratory, where we need to maintain a sterile and quiet environment.
Here you can see the CO2 incubator in which temperature is set at 37 degrees Celsius and below that CO2 level set at 5 degrees Celsius. It has also control buttons, alarms, auto start feature ensuring stable and safe conditions for cell growth.
This is the inside of the CO2 incubator. You can see the stainless steel chamber with multiple shelves, with flasks, petri dishes, and well plates.
So CO2 incubator is the hero behind every big biotech breakthrough.
Without it, no cells, no science, no Nobel Prizes. So respect the incubator.
Now we'll see the do's and don'ts of an incubator. In the part of do's, limit the opening of a door again and again due to maintaining the stable environment conditions.
Sterile regularly to avoid contaminations and also the overflow, Because of that, air conditions and air environment can be disturbed.
Not use incubators as oven or to keep your tiffins inside. Do that at your home. Don't let the incubator open for a longer period of time because it can cause contamination.
Putting harmful and unknown substance inside the incubator as it can be really harmful. The incubator science made easy. And without incubator, gym membership free.
We shake it to the max. Will the DJ please turn up the bass to the max? Please just rewind, let me